Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, welcome to session N of Health and Wellbeing. I'm not sure what N is. Mine says 4, the book says 5. We're going to have 3 very interesting talks related to human health today. And our first talk will be given by Bryan Traynor, who will tell us about ALS. Bryan?
Okay, can everybody hear me? Okay, thank you very much for the invitation to talk to you today. As you may guess from my accent, I'm actually Irish, so if I've never given a brief talk in my life, so if I go over time, just tell me to stay, it's up to David to tell me to stay quiet. Okay, so today I want to talk to you about genome-wide association studies in ALS. Just to give you a brief outline of what I'm going to talk about today, I'm going to give you a brief introduction to ALS, to amyotrophic lateral sclerosis, just for those of us in the room who are not neurologists and who are not physicians. I'm going to give a brief overview as well of the genetics of the disease, both in terms of familial ALS and sporadic ALS. And then I'm going to go on and talk about the genome-wide association studies that have been done, performed in the disease. I'm going to try and sort of give you an overview of the field rather than just concentrating on our most recent study, although I will say that we do have some exciting data that I'm delighted to share with you today. So basically, ALS is a neurodegenerative disease affecting the upper and lower motor neurons of the human body. And I was very struck this morning by our keynote speaker who was talking about that patient with a chordoma who had a survival of 7 years. The median survival for ALS is in fact 2.5 years from symptom onset. And in fact, if you consider that it usually takes the patients about 1 year to be diagnosed with the condition, they will generally be dead, 50% of them will be dead, within 1.5 years after they first see me. Which is pretty striking when you consider that patients just walk in and they say, you know, my thumb is a little bit weak. And you're looking at them and go, yeah, in 1.5 years you're going to be dead. Of course, you don't say that to the patient, but you know that's what's going through your mind. So it gives you an idea of how rapidly progressive this particular disease is.
Quick question.
Yes.
Does Stephen Hawking have ALS?
I'm on TV. Am I allowed to say it? So, here's my diplomatic answer. Steve Hawking has a motor neuron disease. I do not know what type of motor neuron disease it is. Certainly, his name is frequently mentioned to ALS patients as a source of hope. You could be like Stephen Hawking. You could live a long time. There are a percentage of ALS patients who live that length of time, but it is a very, very small percentage. The vast majority die within the time frame that I'm talking about. Okay, so the disease was first described by this gentleman here, Jean-Marie Charcot, in 1878 when he was working at the Salpêtrière Hospital in Paris in France. And in fact, in France, it's still known as Charcot's disease. But of course, in America, it's much more commonly known as Lou Gehrig's disease, after the baseball player who died at the age of 38 from the condition. So I think it's fair to say that after about 130 years, the etiology of the condition is pretty poorly understood. And as I mentioned, the very poor and dramatic and sort of drastic survival rate reflects the fact that we really have very few effective therapies for this condition. Just to show you what it looks like in real life, the disease tends to start in one place, often the hands, and spreads rapidly up the arm or the limb that's involved. You get this dramatic muscle wasting and associated paralysis, which is what we see here. And of course, patients where it affects their legs, they end up with difficulty walking. In fact, this lady, this is in fact a video which I will not show you, but it shows dramatically how difficult she is. She finds it to walk and to maintain her balance. About 1/3 of cases start with symptoms in their mouth, what we call bulbar onset ALS. And these patients manifest with difficulty speaking and difficulty swallowing. So in many cases, they actually end up choking and dying of dehydration and malnutrition, although we do have therapies and symptomatic therapies for that. So in terms of the genetics of ALS, ALS can be roughly divided into two types. It can be divided, just like any other neurodegenerative disease, into familial. In other words, somebody else in the family has the same neurodegenerative disease, and sporadic. In other words, it appears to occur randomly throughout the community with no family history of disease. And as I say, like other neurodegenerative diseases, the split between familial and sporadic is about 5 to 10% familial and about 90 to 95% sporadic. That's just like any other neurodegenerative disease. Now, in terms of the familial ALS, we've had some success in determining what the genetic etiology, the background, the backbone of causing those diseases are. And the main gene that was found was, in fact, SOD1, which was first described back in 1993 by my old boss, Bob Brown. And we now know that it accounts for about 15% of all familial cases and a much smaller proportion of sporadic cases. More recently, through the absolutely amazing work of Virginia Lee and John Trojanowski at the University of Pennsylvania, they identified that this particular protein here, TDP43, is the main component of the inclusions that you see within the neurons. And subsequently, Chris Shaw and his group found mutations within that gene could actually underlie and cause familial ALS. And then more recently, a mutation on the FUS gene on chromosome 16 has been described as a cause of ALS. Now, both familial ALS and both TDP43 and FUS account for about 2 to 3 to 4% of cases. So overall, those three genes account for about a third of cases, maybe a little bit less, of familial ALS cases. There are a whole host of other genes that have been implicated in motor neuron disease diseases. Now notice I say motor neuron diseases, not ALS, because many of these are associated with particular forms of motor neuron disease that don't resemble classical ALS, and anyway they tend to be very rare. So I tend to put them in a separate category. In contrast to familial ALS, I think that we have to say that our understanding of the genetics of sporadic ALS is very poor indeed. Unless, with the exception of what I'm going to show you later, we really have no idea of what is underlying sporadic ALS. But of course, me being biased, I'm a geneticist, I believe that it has a genetic component to it. And that is what basically genome-wide association studies are designed to test. Now, just to orientate you, I'm not so sure as to how many of you here have a genetics background, but just to tell you that up until about 7 years ago, the classical way in which to go after and try and unravel the genetics of a sporadic disease was through what we call the candidate gene search. And basically in the candidate gene search, you pick a gene based on your prior knowledge of the biology of the disease, and you sequence that gene in a series of cases and in a series of controls. And if you find more mutations in cases than you see in controls, you write a paper and you say you think you found a gene that causes it. Now, we now know that this candidate gene approach really does not work very well at all. And that's for a variety of reasons. First and foremost, it's based on the biology that we already know, which means that we'll never learn anything new. Because we're picking our genes based on what we know, we're not going to learn about new pathways, novel pathways. Secondly, there's always another gene to go after. So if you do one gene, there's still 26,000 other genes in the genome once you've finished with that one. So we had to come up with a more efficient way of going after it. And third, we know that the approach simply doesn't work. So if you take the example of schizophrenia, there's a paper published a few years ago in Nature Genetics by a chap called Rudy Tanzi, where what he did is he went back over all of the candidate gene studies that have been published in schizophrenia. And he isolated about 3,500 variants that had all been described as possible causes of schizophrenia. And he followed them up in terms of epidemiological data, followed up the paper, each of these ones in papers, to see how many of them survived replication by other groups and independently. And he found that out of that 3,500 variants that have been described as causes of schizophrenia, only 4 of them were ever actually had sufficient information to really consider them as causes of schizophrenia. So really, that shows you right there how bad an approach candidate gene studies are. And in fact, we often joke that really candidate genes only generate papers. First and foremost, there's the paper showing the candidate gene association. Then there's the paper explaining, failing to replicate it. And then there's the paper explaining where we all went wrong. So it's a very good way of generating papers. And what this slide here is here for is to show you that I'm equally guilty of that. I've published a lot of candidate gene association studies over the years. But really, I think about 5 or 7 years ago, we started to move into the genome-wide association level. And the beauty behind this is that instead of taking one gene, what you're doing is you're searching through the entire genome in a sort of a level playing field. Everything's been tested without any biological basis about the function or knowledge, a priori knowledge about the function. So we started off with this particular study here. This is back in 2005, 2006, when I was actually moving from Boston down to NIH. And this is really about the time that genome-wide associations were just starting. I was very fortunate to be able to access the equipment that had just been installed in the laboratory of neurogenetics. So what we did is we did about 276 ALS cases and 271 controls. We genotyped them on 550K chips. What that does is it basically genotypes about 550,000 SNPs across the genome. Now, even at that stage, we knew that that sample size was very small. But the point behind this was nobody had ever done it before. So we didn't know whether there was a single gene in the genome somewhere that nobody had uncovered that was driving the risk for ALS. Something like akin to the ApoE4 that we know exists for Alzheimer's disease. Now, as you can see from this result, I don't know how many of you here know about Manhattan plots or how to read them, but let me just quickly orientate you. This is what's known as a Manhattan plot, although there are absolutely no skyscrapers here. I'll show you some skyscrapers later. Basically, you've got the different chromosomes arranged here, 1, 2, 3, 4, all the way down to 22. The y-axis shows you the height of the significance. Each SNP that's genotyped and tested is represented by a dot, and the red line represents what we call our threshold for significance, which is corrected for the multiple testing that we do. And as you can see, none of those SNPs achieved Bonferroni significance. I will point out that the added advantage of this particular data set was that it was made publicly available, the entire raw genotype data set through dbGaP. And in fact, the utility of this is highlighted by the fact that it's actually been downloaded and used in all of the other genome-wide association studies that have been published since then. So after that, it became very obvious to us that we were going to have to increase our sample size dramatically in order to detect any loci with an odds ratio of less than 2. And that's what we did here in our second study. This time we took about 500 cases, 2,000 controls, and from that we selected the top 7,600 SNPs and genotyped them in an even larger cohort of about 2,000 cases and 3,000 controls. Again, you'll see that the Manhattan plot is rather disappointing. Nothing achieved Bonferroni significance, though this SNP here on chromosome 7 came close. But unfortunately, and this is a very important thing, in genome-wide association, close is not good enough. You have to cross your Bonferroni threshold, or else really you have to discount it as a causative factor. So this was so we completed that study, just to give you the timeline that I'm talking about. We completed that study about 2008, which was about the time that I was making transition to being the chief of the lab where I am now. And it gave me an opportunity to sit back and to think about how we wanted to proceed, because it was obvious from our own studies and from those published by other groups that ALS was a far more clinically heterogeneous disease than we had previously appreciated. In other words, ALS, the term ALS is not one disease. It's a whole group of diseases that just happen to look like each other. And when you think about it, there are really two ways in which to overcome this heterogeneity. One is to dramatically increase your sample size. And by that, I'm talking thousands upon thousands and thousands of cases. And second is to target, to specifically go after populations which are homogeneous with an increased incidence of the population. So, of course, I wanted to hedge my bets, so we did both things. So we actually adopted both approaches. So I was very proud of the fact that together with Lucy Bruin, I was one of the founding members of the ALS Genomewide Consortium. I say this because I suppose to a certain extent you could say that I kind of took the lazy way out. because we realized that we didn't have the money and we didn't have the resources to collect the several thousand extra cases of ALS, because remember, ALS is a very rare disease, that we would need in order to generate a really high-powered genome-wide association study. So instead, we reached out to all of the other people across the world who had completed genome-wide association studies, brought them to the table in the form of a consortium, and got them to pool their data and analyzed it. And you can see here, these are the different countries that were actually represented in Ireland, England, the Netherlands, Italy, and of course the United States. And in total, we had about 4,000 cases and 5,000 controls. Now, that data, when I say brought them to the table, it was more like kicking and screaming. And of course, every phone conference tends to be like that as well. So this has actually been going on for about 2.5 years now. But finally, we're getting close to having a manuscript. And we do have some very interesting results, including a locus that appears to influence the age at onset. Now, I can't tell you where that locus is because, obviously, we haven't published and I haven't got permission from my colleagues to do this. But I will tell you that a locus affecting age at onset of disease is potentially even more interesting than a locus that affects susceptibility to disease. And let me explain why. If you have a locus that affects just the susceptibility, that would suggest that there's going to be a subset of patients that carry that locus, right? So in other words, if you were to develop a therapy against that locus, against whatever gene it is, it will just be relevant for those patients who carry that particular mutation or that particular set of variants. In contrast, if you have a locus that affects age at onset, what it might represent is the final common pathway involved with the disease. So in other words, what you're finding is that although ALS has many, many different reasons for starting off, they all coalesce onto a final common pathway. And if you can identify what that final common pathway is, you could potentially ameliorate it and alter the rate of progression of the disease. And that's why we're so excited by the fact that we think we found something that affects age at onset. Okay. So just to go on to the main topic of today's talk, which is our study that we did on Finland. So remember what I said, that our second aim was to go after a population where it was homogeneous, a homogeneous population with ALS. Turns out that Finland is ideal for this because it has the highest rate of ALS in the world. It's about 8.2 versus 2.1 in the rest of the world. And furthermore, it's known that Finland has a very genetically homogeneous population. It was founded by 2 men and a dog about 2,000 years ago, and really small population. So it's very, very homogeneous. I shouldn't be saying that on camera because I'm sure my Finnish colleagues won't appreciate it, but we joke about this among ourselves. Furthermore, we know that there's a high prevalence of a particular mutation, a particular allele within the SOD1 gene called the D90A allele that has a particularly high frequency within the Scandinavians. Although I will say that that allele does not account for everything, for that entire increased incidence of ALS. So we did, with funding from Microsoft Research and from the ALS Association, we genotyped about 400 Finnish ALS cases and 500 controls on 370 chips. And here's the results, Manhattan. So this is a true Manhattan plot because we really do see some skyscrapers. First and foremost, we saw this one here on chromosome 21 corresponding to the D90A allele. Now, we had deliberately incorporated those cases, left those cases in our GWAS because we wanted to be able to use it as a sort of a positive control to make sure that we were able to detect it. So we were expecting to see that signal on chromosome 21. We were very surprised, and unexpectedly, we saw this signal here on chromosome 9. Now, this actually is an area that has been previously described in, actually, of all things, Scandinavian families. We only realized that once we went back and looked at the literature. But it became quickly apparent to us that when we delved into it a little bit further and we looked at just split the cases out into those which were familial, had a familial history, versus those who were sporadic, we quickly noticed that these signals were being driven by the 96 familial cases that we had included in our data set. So you can see that the p-values actually increase, the height of the peaks, whereas in contrast in the sporadic peaks, they literally disappeared. They're virtually non-existent. How am I doing for time? So this is a blow-up of the area, zoomed in on chromosome 9. You can see that it really does go up quite high and then goes down back to the baseline once you move out from this region. This locus is basically about 220 kb in size, and it only incorporates 3 genes, namely MOBLIC2B, IFNK, and C9orf72. You can see that within the finish, it forms a very nice block of linkage disequilibrium, as shown here. As I mentioned, it is a previously described area. This is a graph showing the previous linkage studies that have shown this, have been linked into this region. And when you look at these 2 black lines, which is the minimum overlap interval between those familial linkage studies, that's a region of about 7.1 megabases. Our study, in contrast, narrows that down to a region of about 220 kb. So it really is a significant decrease in the locus. And the other important point is that when we went back and looked at this, it turns out that this one locus accounts for nearly 1/3 of all familial ALS cases, which is a phenomenal amount. That's twice as big as SOD1. And we have reason to believe that it is a single mutation. In other words, it is the same thing throughout all of the population, not just in Scandinavia, but in anywhere that the Vikings managed to go on their summer holidays to 1,000 years ago. This mutation traveled with them, and that all around the world, individuals with this particular form of disease all have a single mutation. My problem is, what I work on in the lab is, I don't know what the mutation is. And we've spent a lot of time, a lot of effort looking for that. So in summary, I think that we are finally beginning to see some payoff from all of the money and all of the effort that we've expended on genome-wide association studies. We've identified a locus that through the consortium that influences age at onset, and we've identified, using a honed-in population, a very promising locus on chromosome 9. And I just want to finish by thanking the various collaborators, including David Heckerman, who is instrumental in setting up this particular project. We were very fortunate to have funding from Microsoft Research, without which we would not have been able to complete this. And of course, the ALS Association as well, and my various other collaborators that were instrumental in these studies are labeled here. So I'll open it up for questions at this stage. Thank you.
Questions?
So your work's been based on SNP chips.
That's
How
right.
do you think the advent of sequencing data will change this type of work?
Absolutely. And I actually had another slide in here which has disappeared, which was a picture of a chap standing next to a dam with water shooting out. And that, I think, and I wanted to show that picture because it represented, really it sort of comes from what was being spoken about this morning at the keynote address, that we are being inundated with data. In the past, our problem in genetics was that we couldn't generate the data fast enough. Now, that problem is we're generating it almost too fast. Just to give you an idea, we have one with our brand new sequencer, our HiSeq 2000 from Illumina. We generate about, I would say, about 5 terabytes of data every 10 days. Just to put it into perspective, 5 terabytes of data is equivalent to all of the books in the Library of Congress on disk, which is the world's largest library. And actually, we're after installing just this week a second HiSeq. So that number is going to increase even more than that. So I think that really, you know, this is going to be the thing that's going to face us, the problem that's going to face us, and something that we have to address. It's a good problem to have. I'm not complaining. I want to make that quite clear. This is a good problem to have. It's a very exciting time in the field of genomics, but I think that a lot of stuff has to be ironed out over the next few years first.
So thank you for buying the HiSeq 2000.
Oh, right.
So
2 HiSeq 2000.
excellent. Yes, well, the additional one. So when you look at 9p21, it basically, it sounds like you knock down the noise by looking at the finished population. Is
Yes.
it potential that if you're able to knock down the noise in other populations, you'll see other genes that have the effect?
Yeah, and in fact, we have exactly the same scenario in Sardinia. but was not identified through a genome-wide association. It was actually identified through regular sequencing of known genes. And it turned out that about a third of the cases, the familial cases in Sardinia, are actually due to the same mutation. So, of course, that leaves the other 2/3. So we're really eager to get exome sequencing started on those, because we think that, really targeting these isolated populations seems to be where the money is, and exactly, yeah. So, okay, thank you very much.
Okay. Our next speaker is John Ainsworth, who will tell us about a framework for large-scale modeling of population health. Thanks, John.
Thank you very much, David. Can everyone hear me okay? Good. Well, thank you very much for inviting me to speak at the Microsoft eScience Conference 2010. I am going to talk about a framework for actually doing predictive modeling of the health of populations, both looking at how people become ill, so actually contracting particular disease, and actually how you model the disease itself. And as part of this framework we're developing, we're really interested in answering the questions about where is it best to place the money that's available into healthcare. I mean, that's the whole premise of the thing. And as you can see, almost a cast of thousands working on this, and a variety of different funding sources. So principally the work's been carried out from the Medical Research Council and the National Institute of Health Research by researchers at the University of Manchester and the University of Liverpool, along with some colleagues in some other institutes, the Northwest Institute of Biohealth Informatics and the nascent Northwest eHealth Organisation. So this is really at the very heart of what we're trying to do. It's basically saying, where is the most bang for my buck, if you like? With limited resources for healthcare, how do we know where to spend it? There are plenty of different choices. Do you do public health interventions? Do you do surgical procedures for people when they become sick? With the intent of saying, well, let's provide the best quality of life years for the entire population and make sure the money is spent equally and fairly. This is more particularly important now at this time because within the UK, as you probably know, we have a nationally funded health service, and we're looking at cuts of about 20% from central funding for the health service. So everybody is very, very focused on saying, well, okay, we're going to have less resources, let's spend them more effectively. When I go and talk to my colleagues in the NHS, commissioners who are responsible for planning which services they need and allocating the budgets, and you ask them, say, well, how do you work out what the budget for next year? And you ask them once and they say, well, actually it's 10% more than last year. And you go and ask another 10 people again and they say, well, what's the budget for next year? And they go, 10% for next year. And you start to think, well, I thought they were joking, but maybe they're not actually joking at all. Actually, that's how they do it. So it's not quite as simple as that, but this is basically the model that they use. So you often get a national directive that comes down, which leads to pressure about making budgets. And the evidence they use for making decisions is what can be described as quick and dirty. So going to the grey literature, the white literature, ask those locally who have an opinion. And obviously the effect you get there is the people who shout loudest get the most. Very simple statistics, peer advice, which again kind of, excuse me. So this is the literature that's not actually published in peer review. So it would be data collected from local sources, local technical reports, local analyses. And you synthesize it and you build up a local policy. And then what happens after that is it gets pushed away somewhere, forms part of next year's grey literature, and it drops basically into a black hole and may never ever be seen again. So the ideal solution would be when they need to make a decision, they'll find a model, a policy model, that's relevant to the question they're answering. They will seed that model with parameters from their local population, which actually characterize, for example, age distributions, gender distributions, factors of deprivation about the population on a geographical basis. You want to feed these things into the model. And then indulge in some what-if scenario planning. So basically execute this model many times, play around with what the interventions might be, and examine the effect on the population, in terms of the number of deaths that you might see, the number of people that you make live longer, and also balancing it against the economic costs. So the work I'm reporting on today is basically Impact 2, and we're building on top of the Impact 1 project, which was a policy model for coronary heart disease. And it was actually constructed as a conventional Markov model, actually built into Excel. And it was a complicated model, and it had 44,000 different cells. And actually using it was extremely difficult. You had to have a certain level of expertise, so you had to already climb that enormous steep ramp to actually get up there to start using it and have the confidence to do it. Now, there are problems with spreadsheets as well, which I'm sure lots of people know, in that they're not particularly transparent or easy to understand. They're not amenable to analysis. And indeed, there's been some notable corporate losses through the misapplication of spreadsheets. I think Fannie Mae lost $1.2 billion through misapplication of spreadsheets. And so we said, well, okay, this is not sustainable if you want to do this. We need to build a proper framework where you can actually go in and look at everything that's going on. So it needs to be transparent all the way from the source code underlying it, through the model definitions, through how you fitted the models, all the way up to the simulations that you've run, all the parameters that you've seeded it with. And so that's basically what we aim to do. We also embarked on a survey of existing CHD policy models, and we found 70 different modelling attempts. And out of that set of 70, only 10% actually ever got published as a paper. And if we look at the survival rates, which really indicates how well they're adopted, there was none that survived any longer than 10 years. So we might speculate as to why this is. It's possible that the models actually didn't work very well. It's possible that the source of funding ran out. There are numerous different reasons. So why do we actually, well, what questions do we actually want to answer with our policy model? And principally, there are 5 different questions or categories of questions we want to answer, which is, within a defined population, can we predict how the burden of a disease will change over time? So, for example, if I was starting to look at this maybe 15 years ago and wanted to simulate obesity, that might have been something quite interesting to do and look at the pressure that caused eventually on the health services. If we have certain different interventions or treatments, what will the impact of those interventions and treatments be in terms of quality of life years, life expectancy? Thirdly, if we were to provide interventions actually at the public health level, so if I wanted to invest a lot more money in smoking cessation, what would the impact of that be, say, on the rates of coronary heart disease? In terms of life expectancy, are we better off investing money in prevention rather than treatment? So where does the balance fall? Is it better to treat the entire population, or when somebody does get sick, actually provide effective treatment? Which naturally leads into the second question. Okay, so if we have a population and we can identify those at higher risk, should we actually target those with specific interventions, or should we do a blanket approach across the entire population and actually say, well, actually, we don't know who's really going to get sick? So these are the broad categories of questions we've set out to answer with the simulator. And so what we do is the whole premise is the whole thing is evidence-based. And often the evidence is incomplete, not available, at best maybe patchy. If we had perfect evidence for every intervention across every kind of population, it would be very easy to construct these models, but the fact of the matter is that we don't. So we've actually split the simulations into two. We have what is called the population side, which generates incidence to cases into basically a clinical side model, which actually simulates a particular disease. So the particular disease we're interested in today is coronary heart disease. That's the model I'm going to show. And we put this all into basically a unifying framework where you can actually simulate the competing risks of individuals through probabilistic graphical models and discrete event simulation. So the framework itself is generic. What you have to do is you have to build the model, you have to fit the model, you have to validate the model, and then you can start running simulations. So this is what the CHD model looks like. And as I said, it splits into two sides. We have the incident cases in the population model, and so you'll see all the different risk factors associated with cardiovascular disease risk. And then these feed into a model of coronary heart disease, and we have all the different states associated with coronary heart disease. Now, this model actually comes through a process of evidence, but also expert elicitation. So you go around, you ask the clinicians what they think the different health states are in this model and what the transition probabilities between the different states should be. Interestingly, it's easier to do that side of the model than it is to do this side of the model because the evidence base and the knowledge is much better. When you're talking about public health interventions, actually getting the evidence becomes much, much harder. So what we do is we generate incident cases, we feed them into the CHD model, and you simulate them through discrete event simulations until the population or the period of time for the simulation expires, and you end up with people in either CHD death states or non-CHD death. Just a brief overview of the statistics. So currently, we're generating the incident cases using a standard epidemiological approach. We're not using the Bayesian network approach at the moment, and we have actually, over the last few weeks, generated a new approach, which I'll talk about later. For the CHD simulations, we're actually using the epidemiological data from something called the SLIDE cohort. Then we simulate the life course for each patient using discrete event simulation through the CHD model. And as I said, well, we don't actually have all the data to build a perfect model because it just doesn't fit. It just isn't available. And so what we do is we need to fit the model to the constraints that we have. And to do this, we're using an entropy-based approach using simulated annealing, which enable us to get a fit for the model with the constraints that we provide. And then discrete event simulation throughout the disease model uses a competing hazards framework. So there are various different hazards in different states, and these compete together, and through the process of the simulation, you either fall one way or you fall the other way. Just looking at the software architecture of the system that we've built, it falls into four main functional components. I think it's important to make the point that all of this is transparent, and we're releasing it as open source code. I've seen the artistic license. But the entire system is a web-based system as well. So you can access all of this through your web browser. So there is no need to download any special software or anything. You just go to the site and you can actually start using it. So at the top we have the presentation layer, which is the bit that people see through their web browser. Every user in the system is registered and they have their own user data space. So if they're creating models or running simulations, these are all stored for them. Because of the models and the simulations require quite a lot of data, we have basically a data management function that manages all this data. At the very back end of the system, we actually have something called the engine service, which is actually where the engine simulation runs. And the broker service exists so we can swap in and out different types of engines or put the engine on different hardware platforms. So again, The whole thing is intended to be transparent to model developers, model validators, and model users. So just going through a few screenshots that we have here. So you'll see on the right-hand side is actually the CHD model. And this screen is actually the constraint editor, where you could apply the constraints to the transitions on the edges before you're going to do a model fit. And this is what model fit interface looks like. And you can see you do lots of iterations of simulated annealing, and eventually you start to converge on one particular solution. And if that solution is close enough fit, then you can go with that, or you can make another attempt, and you'll see numerous attempts have been made to do the fitting. In the case of the CHD model, we actually do distinguish between males and females because their characteristics are slightly different. Now, if we go back to actually running simulations, so we have the model on the right-hand side again, and we have the various different states. Now, what we can do here is look at all the interventions. So the interventions are created as part of the model. So when you finish creating a model, you then want to run simulations on it. And when you're running simulations, you're interested in changing how the different interventions are applied. So here we've got basically an uptake editor. So for each age range, for ACE inhibitors here, which is one of the interventions that applies, you can modify the different uptakes in the population. But you can also modify the availability as well. So this means that you're actually controlling how you apply particular interventions to the population. And you see in the CHD model, we've got all sorts of different interventions which don't apply at each and every state in the model, but this one is unstable angina because it's highlighted in red, but these are all the interventions that apply. And so we've got ACE inhibitors, aspirin, let's see, coronary artery bypass graft, and we've got stenting down here, and various different drugs and statins at the bottom. So it's important to say that the system allows you to build any model that you want to and apply any different range of interventions. If you have the knowledge through expert knowledge elicitation or evidence collected from various sources, then you can go to the system and build a model like the CHD model, save it, fit it, and then validate it, and then publish it and make it available for other users to actually run their simulations. So it actually provides the end-to-end process from actually designing the model to actually executing the simulations. And here you'll see, actually, is it a report from a simulation, and you can see it's comparing to, someone's created a baseline simulation, someone's reduced the uptake of statins, and you can see the difference in the deaths in the various different age groups. And as you'll see, as you would expect, the reduction of uptake in statins has resulted in a small increase in CHD deaths, which is probably what you would expect. So the system is available, and you can go to it, and you can use it, and the URL for it is www.healthimpact.org.uk. You just need to register for your account, and then you can start using it. We just look at some of the initial results of validation for the CHD model. And again, this is the output of the model. Simulated is in red. The real data is in blue. And the real data is coming from the slide cohort. You can see that we're starting to get quite a good fit. The model needs to be tweaked and tuned. And actually, validation is a very time-consuming, laborious process. But you can see we're starting to get face validity in terms of the fits we're starting to get. So where we plan to go next, we focus very much on the disease side of the model up to now. Our next step is to look at the population model and actually to be able to start asking the questions about is smoking cessation more effective than statins, if that's where you put the money in. We also do need to put the economic costs in because we actually haven't done that yet. but it should be a fairly straightforward matter. We want to start building models for other diseases, so working with other clinical groups to actually develop these models and validate them. And ultimately, we want to link to other projects that we have, most notably the healthcare e-labs through Northwest eHealth. So this is a nice shot of some early work we've done on the population side model, which is called the stock of health approach. It actually follows an accelerated failure time model. And we've actually developed this so far in Microsoft F#, which is proved actually to be very effective in terms of development time, but also in the way that it scales. And you can see a snapshot of it running on a multi-core machine, and without having to write any extra code, it's nicely balanced across all the cores. So that's important for us, because when we make the simulations more complex and you increase the population size, the computational requirements shoot right up. Part of the work that's going on in Manchester, and actually is following, is actually part of what I picked up as the theme of this conference, is actually sharing work, exporting work, publishing it, make it more available. And so you're able to export the simulations and the results of simulations as something called an eLab work object, which is actually a collection of all the work and all the notes and all the workings and all the methods that you actually do inside an eLab. An eLab is very much a concept that we generated around Manchester, whereby you're basically providing an in silico workbench in which you can carry out these types of studies. And this is some of the work we're doing with the Northwest eHealth group, which is the NHS eLab, which is really a layer that sits on top of the various clinical systems, electronic health record systems, and actually brings them all together, applies the appropriate governance, gives guarantees of confidentiality and privacy to patients, and actually opens up the records for various different uses, such as clinical audit, for feeding data into these population models. There are numerous different applications that you can apply the observed data to. So in conclusion, I think we're actually starting to show and starting to convince people that actually you can do a lot better than just sticking your finger in the air and making a guess. One of the key things is to ensure the transparency, and it's to generate a culture of sharing and improving the models through iterative development. Again, I said a lot of the evidence that goes into this is not necessarily evidence, but expert knowledge. And yes, they're complex, but hopefully we're trying to make it so that the complexity is exposed in a transparent and understandable way. Thank you very much.
Thanks, John. Questions? Jen was first. Just pass the microphone.
It looked like you were validating your model by showing that the synthetically generated data gave the same death statistics as the real data. I'm just wondering if you did any sort of holdout experiments where you could actually train your model on part of the real data and show that you can predict aspects of the real data, because I think if you don't do that, you have the potential to seriously overfit your model.
Yeah, so we are going through that process right now.
Okay.
Hello, my name is Chris van der Meijden. I'm from the veterinarian faculty of the Ludwig Maximilians University in Munich. And I really enjoyed your talk, and I was wondering if you ever tested the universability of your framework in veterinarian medicine. I'm thinking about avian flu. Can you easily change the values for a lifespan of the species?
Yep, so anything that you see there in the model, you can actually define yourself. So as long as you can define a health states model, you can draw the nodes, link the edges together, and then put in all the competing risks in the different nodes, and then play around with the interventions. So yes, if you're asking about whether it's a model for communicable disease, I think the answer is no.
I actually have the same question in sort of a different guise. Obviously, you're working within the context of the NHS. And it's not just in the NHS that healthcare costs are spiraling out of control.
Yes.
The US is also a particular area where this is like, you can't pick up a newspaper without seeing it. How applicable is your modeling to the US healthcare system, which is, and I've experienced both of them, they're radically different healthcare systems. So how applicable is it?
Leaving aside economic costs for the moment, just looking at the effects of various different interventions is not a problem because, as I said, you're able to build your own model, apply your different interventions, you're able to adjust the populations that you actually simulate in terms of age, sex, deprivation, all sorts of different profiles. So in that sense, it's a generally applicable model. I don't know enough about the healthcare system in the US to actually comment in terms of the payer-provider split, how it would actually work out. So I'm kind of sidestepping your question, I'm afraid.
I mean, it might be applicable to Medicare and Medicaid, which are government-run insurance programs.
Yeah.
And where they have, or they're developing a structure to do what you're talking about. I'm just wondering in the larger context of healthcare in America, which is really, because interesting enough, when you look at it, Medicare and Medicaid are actually very effective in terms of cost containment.
Yeah.
It's the other areas that are really driving it.
Yeah.
Thank you. That was a really nice presentation. I'm trying to grasp how you're saying this is a discrete event simulation model, but it seems more like at least if I'm understanding what you did, it's more like a sort of a stochastic decision analytic model. You just have people, basic representation of people moving through decisions and then stochastically deciding which decision tree they go through. Am I understanding the model correctly, or am I missing something?
Well, I'm not a statistician, so the modelling side of it is not my thing. The software side of it is my thing. But essentially, you define the model, so there are various different states. And it's basically a proportional hazards model. So at each state, you have various different risks that compete against each other. And yes, when you start a simulation, you basically take an individual and then you run them through the model and through the different states and through the different branch points that they may take until they die.
Great. Okay. Our last speaker will be Gill Bejerano, who will tell us about generating functional hypotheses from genome-wide measurements of mammalian Cis-Regulation.
Okay. Thank you very much, David. So it's a pleasure to be here today. And I appreciate very much the invitation. And I will tell you about, well, can I control the volume? Or? Yes, you control. All right. So I'll tell you about a tool that we have developed really that addresses analysis of next-gen sequencing data in ways that are very relevant. And I'll walk you through what we've done and how we've done it. But first, I'll give you a very brief introduction to the way my lab, which is shared between developmental biology and computer science at Stanford, looks at the genome and tries to understand it. So if you look at a human being, there's something on the order of 10 to the 13 different cells in an adult human being. And of course, we all know that all these cells have essentially, to a first approximation, exactly the same genome. And in that genome, these cells encode between 20, maybe 25,000 protein coding genes. But if you look at different cells in our body actually express very different repertoires of these genes. And they use this expression, this turning on and off of expression, as a means of defining themselves as very different cells, as a means of communicating with each other, et cetera, et cetera. So one of really the key revelations that have come out of the genome sequencing area of the last 10 years is an appreciation, a growing appreciation, I would say, of how much of our genome is actually devoted to this, what we fondly call the control layer of the genome, turning genes on and off in exquisite patterns. I'll give you a taste of that in a minute, during development and later on. Okay, so anecdotally, the mass of sequence thought to encode gene regulation is now thought to be at least twice as much as the mass of genome thought to encode how to exactly make a protein coding gene. So you should think of this, the green boxes here, as the how to make a protein, and the red boxes as to when and where and how much. That's the control layer. So I thought I'll give you just a brief view of what the real data looks like. So this, if you focus just on the blue labels here on the top, so this is a snapshot from the UCSC genome browser, about 9 million bases from the human genome, and every label here is a different protein coding genes. And one of the things people have noticed for a long time is these regions that have come to be termed as gene deserts, where for a whole megabase, there's not a single gene in there. So these were thought to be very boring regions, and there were actually a lot of discussion initially of whether they should even be sequenced or not, if there's nothing interesting in them. And recently, though, as I told you, with this genomic revolution of seeing whole genomes, the one thing we can do is we can align this region in the human genome to the same region in other species and ask where is it that there are DNA regions that are clearly evolving under purifying selection between different species. So if they evolve much slower than the neutral rate, you would say they're under purifying selection and therefore likely have a function. So the first thing I'm showing you here is, the mass of what we call conserved and non-coding sequence in the human genome. So this is, again, it's just different layers of annotation over the same 9 million bases. So every tick mark here you should think of is between 100 and 1,000 bases that are clearly evolving under purifying selection between different species. And this is where they localize in the human genome. So you can see here that these gene deserts, many of them, and this is really representative of the human genome, are essentially chock full of this function, which is not protein coding function. And what we've really come to realize, I'll show you just another snippet of proof of that, if you like, in a second, is that now we think of these not so much as deserts of genes, but actually as regulatory jungles. So as regions of the genome that are chock full of these sequences that tell the genes next door exactly when and where and how much to be expressed. And really, one other circumstantial evidence, if you like, before we jump into the actual proof here, is if you look up from these regulatory jungles into the genes that are found next to them, you invariably find genes like these 3 Iroquois family genes. So these are genes that play critical roles, or the proteins that they encode for, play multiple critical roles during early development. Okay, and if you think, how can you make one gene play critical roles in different contexts of the developing embryo, in different tissues at different time points, you exactly need that battery of genomic control. Okay, so here's just a sense of quote-unquote proof. You can take these regions, and people have marched through 2,000 of these regions so far, roughly. So here's just one region of several hundred bases. And you can essentially, if you think this genomic region regulates that gene, you make a copy of it outside of the genome, put it in front of a reporter gene, inject that into a one-cell mouse embryo, and assay the transgenic embryo that now contains your construct for the expression of that gene. So you essentially ask, where can this element drive the expression of this gene? And here you see one such element that drives beautiful expression in the forebrain only at embryonic day 11.5 of mouse, etc. So over half of these regions that have been tested already in an arbitrary time point give you these exquisite patterns. And essentially what we see is that if you have a key gene here, in this case the SAL1 gene, but what matters here is the concept. So this is a gene that if you have mutations in human, you actually have defects in brain development, in limb development, and in neural tube development. And here you see that at this stage of mouse development, this is exactly where the gene is expressed. Okay, now if you go around and you search around the gene, you would find the switches I was talking about. So here is a switch, again, empirical data, where this switch will drive any gene next to it in the forebrain, another one in the neural tube, and a third one in the limb. So you can think of it as the union of the expression patterns of these guys make that gene turn on in a very specific pattern. Okay? So this really understanding of the genome is opening up a lot of avenues for us, and I'll give you just a taste of it, I think, very briefly now, because I want to tell you an actual tool story in this case. So what my lab is focused on is really on understanding how what is the contribution of that regulatory layer, like I said, which has been very poorly appreciated through nobody's fault. It was essentially invisible before whole genomes were sequenced. So these are regions that can lie up to a whole megabase upstream or downstream of the gene they regulate. And people essentially will only focus on the coding genes and maybe, the 1 5 kb promoter region upstream immediately. So we care for how these regions actually pattern embryonic development, and we focus on 3 tissues in particular, the limb, the forebrain, and the placenta. We care for how these regions have played a role during human evolution, more recent human evolution, and going further back in time. Again, these are questions that have been studied very carefully in the context of genes and continue to be studied. But if you ask that about the control layer, you have a whole new regime to work with and beautiful stories to appreciate. And obviously, if you think of this as when things go right, this is the side where things go wrong. And this is where you can imagine patients, and we have by now documented cases in different phenotypes where the genes of the patient are perfectly fine. So you can code for the protein at the right time, at the right place, if only you had the regulatory sequence not messed up. So if these regions are affected, even if the gene is fine, you get the wrong control. So they turn on for too long, or they never turn on, et cetera, et cetera. So we're trying to characterize patients whose genes are perfectly fine, but we find what's wrong in them, and what's wrong in them is this regulation layer. Okay, and then the lab tries to do, I try to keep this nice mix between people who are computational, people who are experimental, and people who try to straddle across, and that makes for very nice interactions and very nice collaborations within and between the lab and other labs. So let me close the circle for you for a second for those who are less familiar. So if 20 to 25,000 protein coding genes are encoded by our genome, and they can make proteins at will, about 10% of these proteins go back and bind the genome. So they come back, but they bind very specific DNA sequence. Every one of these so-called transcription factor binds specific sequences such that the way we currently understand one of these switches, of which, as I told you, we estimate now there may be about 1 million, if not more than that, a switch here is a key set of DNA bases such that essentially you have a landing pad for a subset of these proteins. And only a cell that expresses the right set of proteins would have all of them come in here together and activate this switch. So you can think of it as an AND gate if you come from the more computer science side of things. You need these 3 in there. They activate the switch together, add another gene, another protein, so to the pool. And if that protein itself binds DNA, well, you can imagine how the state continues to change. That's how we understand the switches. Okay, so if we now are at a stage where we can try to understand these codes. We want to understand where the boxes are, and we want to understand how they encode for this beautiful tissue specificity, tissue time point specificity that I showed you. And one of the exciting experiments to be enabled by whole genome sequencing is the following. Instead of sequencing a whole genome, you can sequence a select subset of the genome. So in this case, you take an antibody to a specific transcription factor, and essentially what you do is you sequence all, potentially, and only the regions bound by your transcription factor of interest at a given populations of cells. So essentially, you look for all the places where this green guy is bound to the genome. So this is known as ChIP-seq, or chromatin immunoprecipitation, followed by deep sequencing. And what you get at the end of the day is a series of locations in the genome you've measured where the key transcription factor you looked at is bound. Okay? So the question then is, what did I learn about transcription regulation by performing this assay? So for the first time I can do it, but how can I interpret it? So what people do is the following. One very nice compendium of knowledge is people have gone and collected different functions of intracellular processes, extracellular processes, et cetera. And they would say things like, to build the actin cytoskeleton of a cell, you need this gene and that gene and that gene and that gene and that gene. To do this and that signal transduction, well, these are the genes who play a role in there. So one of the things you can ask is whether the transcription factor you just assayed is enriched for directly regulating a larger than expected subset of genes from any particular function. And if it does, then you have a handle on a subset of the binding sites. And if that is a function that is of interest to you, you can go and further pursue that by chasing down individual elements, understanding their function, understanding their disease and evolution aspect, if you want. Okay? So I think what I like to do is I'll walk you through a live example from the literature. So here's one transcription factor. The serum response factor has been assayed using ChIP-seq. And at the end of the day, the original authors had 2,400 peaks in the genome. Now, they wanted to make sense of these peaks. Now, they had prior knowledge that this gene, the SRF gene, is a master regulator of the actin cytoskeleton. So you would expect to find this gene regulating a large set of genes that are used to build the actin cytoskeleton. That's part of what it's known to do. Okay? So let me show you how these experiments, how these enrichment tests are essentially done right now. So essentially what people do is the following. They have the set of ChIP-seq peaks. They have the set of genes in the genome, which are the arrows here pointing in this or that direction. And they take some term, in this case, actin cytoskeleton. And then you color all the genes that actually are involved in actin cytoskeleton formation. So you have the genes that are involved and the genes that are not. The next thing you do is pretty much everybody ignores distal binding events. And I'll show you in a minute why it's not trivial to take them into account. So for various reasons, if it's not up to 2 kb away from the gene that it regulates, let's just ignore these. And then you're left with only the things that are proximal to the gene starts. And those you convert from binding events into a subset of genes. So the whole test is done on genes. These are like the two things that are essentially, quote unquote, wrong here. I'll show you what I mean by wrong. So one is you ignore the distal binding events, and the other one is you convert a set of binding events to a list of genes. So in this particular example, in this toy genome I have here, there are 8 genes in my genome. 3 of them are tied to actin cytoskeleton. I have picked 2 genes only, and one of these is associated with actin cytoskeleton, and I can do a hypergeometric test to ask how exciting is that thing. Okay? So what's the problem here? So there's a huge advantage to doing that. And the advantage is if you have a small list of genes, there are many, many tools that will tell you what is interesting in a list of genes you came up with. Why are all these tools out there? Because these tools were essentially designed to the previous wave of innovation, and that was microarrays. So microarrays really measure at the level of a gene whether a gene goes up or a gene goes down. And the list you get at the end of the day is how a gene behaves. Is a gene like this or is a gene like that? So the unit of measure is really a gene. Problem is, and I'll show you that in a minute, is that when you try to force this next generation data, which is whole genome, through these tools, you end up missing a lot of things. So again, the good thing for people who are users and are driven by the questions is I need a tool to do what I want to do. These were the tools that are available. And now I'll show you what happens from a tool builder perspective when you actually build the right tool. Okay? So let's continue with the original story of the SRF. So these, if you do the test the way I showed you a minute ago, look only at the proximal binding events, convert into a gene list. These are the terms that are enriched from the gene ontology, one of the largest and best annotated ontologies out there, probably the best. These are extremely general terms. So what did you learn here? You learned that this gene is found both in the cytoplasm, in the nucleus, and it's involved in some form of transcription. That's it. So you go back to the original paper in Nature Methods, and you see that the authors would say, well, you know, what we can learn from our data is that SRF is involved in basic cellular processes, particularly those related to gene expression, right? But you wanted to find the actin cytoskeleton. This is what you know this gene is doing. So if you keep digging down this list, there will be an actin cytoskeleton term. It's ranked 28, and its p-value is way, way lower than all of these. So the first question you can ask is the signal not in my data? What am I missing here? So let me show you 2 things here. The first thing to note is that by taking only the proximal binding events, you miss out on a lot of the signals. So remember, this is genome-wide, right? You sequence whatever's out there, and even though you are interested only in the proximal promoters, you let the thing run loose, and I'll actually show that that's very fortunate. So here I'm binning every single binding event for 8 different ChIP-seq data sets, how far is it from the nearest gene? Up to 2 kb away, which is what most people take, 2 to 5, 5 to 50, 50 to 500. This is from the nearest gene transcription start set. So you can see that people would throw out anything between 30% and 90% of their signal by only looking at proximal binding events. So the first thing you say is, oh, okay, if that is the case, let me get all this signal back in there, and let's see if it matters. Okay, so does it matter? The first thing you can do is you can say, let's not throw the distal events. Let's assign every binding site to the nearest gene. Maybe, no matter how far it is a bit queasy. Let's say if it's up to a megabase away from the nearest gene, let's assign it to that gene. Now, when you do something like that, you get different size regulatory domains to different genes. And the problem there is exactly what I told you before. There are particular categories of genes who like or who need, quote unquote, the large jungles next to them. So they have larger basins of attractions than other categories of genes, presumably because they need all this cis-regulation in them. So if you take, for example, multicellular organismal development, again, I told you this is a developmental story, 14% of genes are tagged for that, but really 33% of the genome gets painted for that attribute if you just associate everything with the nearest gene. So what happens then is you can forget about the SRF ChIP-seq set for a second. Just take a random set of 2,000 peaks, throw it at the genome. You don't want to see anything when you throw a random set of 2,000 peaks at the genome. And what you get are beautiful, beautiful Bonferroni corrected p-values for, well, exactly the terms of the type of genes that need the large regulatory jungles next to them. So this is bad, right? You want to draw in these distal things, but this is all this is false positive, right? You have an absolutely random set of 2,000 peaks. So the next thing we did is the following. We said, look, this is an assay that is done over binding events over the genome. You should not convert this to a gene list. So the next thing we did is the following. We said, look, we'll do exactly the same as before, associate each gene with a variable length regulatory domain. And then essentially, we are going to forget not about the binding peaks, we're going to forget about the genes. We will say, here is the genome, here is how much of the genome is associated with actin cytoskeleton, and every peak that lands there is a success, and every peak that lands outside of this set of regulatory domains is, quote unquote, a failure. So in this case, now you have 33% chance, as you should, of hitting a region related with that. You've thrown 6 ChIP-seq peaks, and 5 of them hit the annotation. And this is a binomial test, and you run that. So as I'll show you, you can only get a sense that for false positives, this is excellent, because you essentially have, on average, no false positives. If you throw a random set at the genome, roughly 33% of it would hit these regions, and you would be absolutely not interested in anything that comes out of that. That is excellent. I'll show you in a minute what happens in the good set. So we've termed that the genomic regions as opposed to genes, enrichment of annotations tools. The tools section will come in a second.
Question?
Please.
How do you know which regions were associated with actin cytoskeleton?
Well, so the genes come from these prescribed lists, right? And the next slide, we say we'll make the following observation. So in future, we can actually measure the regulatory domain of each gene experimentally. We cannot do it now, but the tool is essentially set up for that already. All you have to do is tell me what the regulatory domain is. So what we do now is we essentially determine that computationally. And the way we like to do it is every gene gets 5 kb upstream, 1 kb downstream, and then is extended up to a megabase away or until it hits the regulatory domain of the gene upstream or downstream. That is the way we do it. And in fact, in the tool, we allow the users to play with that as much as they like. But notice that there's a healthy balance here, so we may not get all of them right, but as I'll show you in a second, we get an overwhelmingly large fraction of them apparently right. And this healthy balance between the longer the regulatory domains, the harder it is to get excited by the null hypothesis, really makes that useful. So let me show you essentially what the results look like. Here's what happens when you try to force your set through a microarray-based approach. And when you use our approach, these are essentially the terms that come up to the top, exactly the terms you want to see. Actin cytoskeleton, actin binding, in every case, we can substantiate those from the literature. But essentially, that says that, in the literature, SRF is known to regulate 5 genes. How? Typically is unknown. Here we tell you there are 30 genes regulated by SRF, likely through these exact peaks. And you can go and follow these peaks. And we can go on and on, find pathways that SRF is involved in. Again, substantiate a fraction of that through the literature, gene families that it regulates, targets of other genes that it co-regulates with, et cetera, et cetera. We've gone on and that out for several transcription factors and transcription-associated factors. And really, when you respect the tests that you're doing, the results come out the way you hope them to be. So that's been very useful. What we've done then is we went and curated a large body of literature. We put on our computer science hats, and we optimized the way we do the different tests. And at the end of the day, we can do it so fast that we can essentially just put it out on the web with a single machine, fairly strong one, but not a huge one and not cloud or anything like that. And essentially, all you have to do is come and tell us which genome, what is your set of peaks, and that's it. You're set to go. So this is what the results look like. You get some fancy controls, but essentially it tells you enrich for actin binding, actin cytoskeleton, etc. You can, as I told you, there's a nice set of controls. You can go in and dig through any one of these terms. If you care for this term, you get exactly a set of peaks, the set of genes, the definition of the term. And if you want to, you can jump into the UCSC browser directly, see what things look like, and design the next experiment for genes that you care for from that subset. And then we've also worked with UCSC, who I'm good friends with, and I spent a bunch of time with before starting at Stanford. So essentially, you can go to UCSC Genome Browser and pick any set you like. In this case, the ultra-conserved set I worked on before, and just say, send that to GREAT, hit submit, and that's it. You're there. So the direction is bidirectional. And this is very well documented, and people can also come and inquire with us. You just send us a URL, and the results come up. So this is nice. You can export that, et cetera, et cetera. I'll just show you how the tool is catching on. So one of the things we want to see is, there's so many tools out there. You really want to see that the community catches on to it. So these are just page requests on a daily basis. These are how many users we have and how many of them are repeating. You can see that, some lectures we actually give a lot of new people coming in, et cetera, et cetera. These are the number of jobs that are submitted through the different resources. And this is just from the last 500 entries, some kind of distribution across the world. So the tool is really catching on, and we're very excited by that. So in summary, I've told you about how current technologies are really allowing us to look at the cis-regulatory landscape like we've never looked at it before, and how the tool that we came up with really allows you to understand that landscape much better than if you force it through tools that were really not designed for that. There's an online tool available. We're just QAing the next version, which will be even more exciting than the one we have right now. And really, if you think about it, there is no particular reason to do this only on cheap-seq data sets. So whenever you have a set of genomic regions that you think has a large component that is this, you can push that exactly through the same tool. So this is true for looking at chromatin markers, certain types of them. For example, the ones that are thought to be related to enhancers. You can take a set of those and push them through. You can look at genome-wide association studies that we heard about earlier. So you can take your first 500 peaks from the Manhattan plot that we heard about earlier, and even though the 500 may not be significant by themselves, you can throw them at this tool and ask whether there's any pathway or any function that suddenly becomes enriched through this, and now you get a systems view of what is actually happening in your top-ranking SNPs, even though individually they're not significant. You can do this for comparative genomic set, like the ultra-conserved set that I alluded to before, etc. So let me wrap up by saying thanks to the people who developed GREAT, which is an ongoing project in the lab. And I'll stop by acknowledging members of the lab, particularly Corey and Aaron, who led the project. Acknowledge my funding. Microsoft has been very generous. I'm a Microsoft faculty fellow, and that's been probably the nicest source of money that has completely no ties, which is awesome to utilize at need. And I think I'll stop there and take questions.
One quick question from me.
Sorry about that. Yeah.
This rule of defining regions as 5 KB upstream, 1 KB downstream, and then extending 1 megabase up to the nearest gene. Are you saying that you modified those criteria on a region by region or gene by gene basis by hand curating various sources of information?
Right.
Or is that a hard rule?
So let me, well, actually, we're running behind. So first of all, in the tool, you can go in, and there's a slide that I tossed out, and you can define the regulatory domains whichever way you like. You don't want 5 and 1, you want 1 and 10, or you want to play with larger thing. You can do whatever you want because it's fast enough to do whatever you like.
I see.
Now, what we've done on top is, so that set is computed on the fly, right, computationally, just by knowing the gene transcription start site.
Right.
What we've done on top, just as a proof of principle, is we've taken, a dozen cases from the literature where it is known what the model is, and the model is not like that, just to show that we can do it, and we added those. So there are places where half of a Hox cluster is co-regulated by this one massive region.
Okay.
We toss those in, right? So as far as we're concerned, we just need to know where the green areas are. Mm-hmm. They don't even have to be contiguous, and we're in the game.
Can the user submit a predefined region then?
If the user asks nicely enough.
Okay.
Yeah. Maybe nicely is not the right word. There's no conceptual reason not to do that, right? It's just a question of how easy is it to get that to the web, how much we need to bicker with them if it's wrong, and if not, it just comes in easily.
Yeah. Very good. Other questions? Okay, let's thank Gill again.
Okay. Thanks a lot.